Welcome everyone to Biochemistry Live. I'm Bio, and she's Chemistry. And we're your hosts for this evening. We're going to begin tonight's show with our favorite segment, Mean Tweets. If you don't remember what Mean Tweets it's all about, it's a segment that has some of the most well-known biochemists of our times reading some not-so-nice tweets about them.
All right, so today's topic is mass spectrometry.
Ooh, sounds exciting.
I know, sounds fancy. So our guests today are going to be biochemists who use protein mass spectrometry on a daily basis, as well as some of the people who are really big defenders of it. And some of them are not. So let me tell you a little bit more about protein mass spec. So it's actually an instrumental technique, and that basically tells you the molecular weight of a specific protein.
Fancy.
This process allows for a broad range of masses to be analyzed, and it's quite accurate, to be totally honest. Pretty high accuracy. So in a mass spectrometer, a molecule is vaporized and ionized by bombardment with a beam of high energy electrons.
Whoa, that's really cool.
For proteins, the two ionization techniques commonly used are electrospray ionization and matrix-assisted laser deabsorption and ionization. So there's actually two approaches to categorizing these proteins. And in the first, we take the whole protein, it's intact, we ionize it by either of these two techniques that I mentioned, and then we introduce it into a mass analyzer. And the second way that we can do this is that we take the proteins and we enzymatically digest them into smaller pieces using a protease called trypsin.
Whoa, I've heard about trypsin. I took Biochemistry 1.
Oh, wow.
Yeah, that's really cool.
Okay, so afterwards, these peptides are introduced into the mass spectrometer, and identified by tandem mass spectrometry. So after this, we actually have two more things that go through, or we can do. One of them is isoelectric focusing, and that separates the proteins by pi. Do you know what pi is, Bio?
When I think it's when the charge on an amino acid on the proteins are 0.
I'm pretty sure you're correct.
Oh, well, thank you.
And another way that we have is SDS page, and that's basically gel electrophoresis with a polyacrylamide gel. And through SDS page, we separate the protein pieces, or the proteins, by molecular weight.
Whoa, fancy.
Yeah. All right.
Wow, chemistry. There's so much to protein mass spec.
Oh, yes, there is, and there's so much more. Okay, like I mentioned before, one way of figuring stuff out through mass spec is tandem mass spec, and that's a method for identifying proteins, which, as you know, is the ultimate purpose. Let me tell you a little bit more about it. So at the end of using tandem mass spec, we get a set of fragments, and that is derived from a specific peptide ion. And how we get that is actually because we have we induce a lot of collisions, and they lead to dissociation. Okay, so let me tell you a little bit more. So the fragmentation process gives rise to cleavage products, and those cleavages are around along peptide bonds, and because of this, it's pretty easy,
Yeah.
Sounds pretty
easy,
right?
Yeah.
So because of that, it's really easy to compare our observed fragment masses to a database of already predicted masses for one of many given peptide sequences.
Wow, that is an incredible explanation, Chemistry. Thank you so much.
Thank you so much, Bio. But I think right now, you've had enough of us talking and teaching you about mass spec. Time to see what other people have said about mass spec, and many of them are going to be mean tweets. Let's hear about people's experiences. Go.
Dear Drs. Karras, Hellenkamp, Tonka, Dole, and Fenn, did you really not realize that proteins are part of a complex biological mixture? Love? Thanks, but no thanks.
Yeah.
Tried to do protein mass spec yesterday in lab. Realized pretty soon it wasn't about to happen. Looks like for ionization you need to have equal amounts of the constituents. No, I'm not just kidding. Please do find me some biological samples with the same amount of proteins present. Listen to this. Gave protein mass spec a chance. Really thought I would get some great results. I used electrospray to ionize my sample and pretty much only got 2 signals. At least now I know what I have a lot of. Thanks, I guess?
Go.
So, let's read it. Conducted mass spec for my protein. Did the procedure right and everything. Too bad I can't interpret any of these results at all. The number of mixture components this analysis is trying to show me is more overwhelming than my room with all the clothes thrown on the floor.
LOL at enzymatic digestion. Thank you for so freaking many peptide products. That totally helps me with cleaning up my data so much.
So it looks like there have been quite a few problems with protein mass spectrometry. To help answer these questions, we have a special guest here today. Everyone, please welcome Dr. Protein. Welcome, Dr. Protein.
Thank you for having me.
Yeah, no problem. So how do you respond to all these problems everyone's been having with protein mass spectrometry?
Okay, so before I begin, just want to let you know, this technique is very valid. It's very helpful, very useful.
Mm-hmm.
We do have a couple of problems though, and I'll admit it. We do have a couple of problems, and I guess I'm here today to explain why people are having the issues that they're having and why they're so upset about it, as we so heard. But they were quite mean. I mean, we still have pretty good results.
Yeah.
So let me tell you what the underlying cause of these is. So proteins, they usually exist in a complex mixture. So in a biological sample, you really don't have just like one protein. You have a mixture of proteins and a mixture of molecules, and that's quite a complex, sample.
Mm-hmm.
So that leads to 2 problems. The first of which is related to the 2 ionization techniques that your co-host chemistry did.
Oh, she did a fantastic job on it.
Explaining.
Yes.
Those 2 are electrospray and MALDI. So what happens is that in order to use these two ionization techniques, we need to have relatively equal amounts of the protein that we're trying to analyze. And that's really not possible in biological samples. Different proteins tend to exist in varying amounts in whichever sample that we're trying to analyze. And there's another problem as well related to the same thing. The more of a species that we have, the more abundant that one is, the more likely it is to suppress or drown the other smaller amounts of other species that we have. So that's another problem related to that one. And our second problem, like I said, we only have a couple of problems. Our second problem is that mass spec is a little bit difficult to interpret. What happens is that the biological sample you're trying to analyze is so complex that we get an overwhelming number of components. And one final thing I want to say is that this is really heightened by enzymatic digestion because we get a large number of peptide products. So that's all there really is. Only 2 problems, just a couple. You don't need to be so offended, just a couple. But yeah.
Well, thank you, Dr. Protein. I think you've done a fantastic job in explaining the problems that exist with protein mass spectrometry.
Thank you very much for having me.
Well, thank you for your time. And after the break, we'll be talking to Dr. Ramachandran about Ramachandran plots.